Okay, now next thing, the electromagnetic spectrum. This is basically physics, but let me tell you it in short. How is resolution linked with the nature of light? One of the properties of light is that it travels in waves. The length of the waves visible light varies, ranging from about 400 nanometer to about 700 nanometer. The human eye can distinguish between these different wavelengths. Wavelengths is basically from one trough to trough or from one crest to crest. The length, you learn it in physics. This is not in the sector of biology. The human eye can distinguish between these different wavelengths. And in the brain, the differences are converted to color differences. Color is an invention of the brain. The whole range of different wavelengths is called the electromagnetic spectrum. Visible light is only one part of the spectrum. Figure 1.11 shows some of the parts of the electromagnetic spectrum. The longer the waves, the lower their frequency. All the waves travel at the same speed, so imagine them passing shorter waves pass at higher frequency. In theory, there is no limit to how short or how long the waves can be. Wavelength changes with energy. The greater the energy, the shorter the wavelength, the higher the frequency. Now look at figure 1.12, which shows a mitochondrion. Some very small cell organelles called ribosomes and light of 400 nanometer wavelength, the shortest visible wavelength. The mitochondrion is large enough to interfere with the light waves. However, the ribosomes are far too small to have any effect on the light waves. The general rule is that the limit of resolution is about 1/2 the wavelength of the radiation used to view the specimen. In other words, if an object is any smaller than half of the wavelength of the radiation used to view, it cannot be seen separately from nearby objects. This means that the best resolution that can be obtained using a light microscope that uses visible light is 200 nanometers, since the shortest wavelength of visible light is 400 nanometers. In practice, this corresponds to a maximum useful magnification of about 1500 times. Ribosomes are approximately 25 nanometer in diameter and can therefore never be seen using a light microscope. What does it mean? If an object is transparent, it will allow light waves to pass through and therefore will still not be visible. This is why many biological structures have to be stained before they can be seen. So think about it. This is the light waves and this is the wavelength. See, from one trough to trough or one crest to crest is 400 nanometers. Okay, now look, the mitochondria is enough big to be interfered with the light waves. That's why we can see it. But the ribosomes are far too small to be interfered with the light waves. That's why we cannot see it. This is basically physics, but you need to have a normal knowledge about it. Okay, so the electron microscope. Biologists faced the problem that they would never see anything smaller than 200 nanometers using the light microscope. Realized that the only solution would be to use radiation of shorter wavelength than light. So we have explained why light microscope has limitations in the size of the organelles that we can see. If you study figure 1.11, you'll see that the ultraviolet light, or better still, X-rays, look like possible candidate because they have shorter wavelengths. Both UV and X-ray microscopes have been built, the latter with little success, partly because of the difficulty of focusing X-ray. A much better solution was to use electrons. Electrons are negatively charged particles which orbit the nucleus of an atom. When metals become very hot, some of its electrons gain so much energy that they escape from their orbits like a rocket escaping from the Earth's gravity. It's like that the heat energy is converted into the kinetic energy of the electrons, and so they gain so much kinetic energy that they leave the orbits and they just they just goes out of the orbit like a rocket very fast. And electrons travel in a wave-like manner. You have learned this in your physics, wave particle duality, and so I won't explain you because we are biologists. We do not need to know why it's happening. We are just going to know a bit that how it is happening, just a bit, not that we need it. Electrons are negatively charged particles which orbits the nucleus of an atom. When a metal becomes very hot, some of its electrons gain so much energy that they escape from their orbits like a rocket escaping from the Earth's gravity. Free electrons behave like electromagnetic radiation. They have a very short wavelength. The greater the energy, the shorter the wavelength. Electrons are very suitable form of radiation for microscopy for two major reasons. Firstly, their wavelength is extremely short, at least as short as that of X-ray. Second, because they are negatively charged, they can be focused easily using electromagnets. A magnet can be used to alter the path of a beam, the equivalent of a glass lens bending. Like, we do not know why this is happening because we are not students of physics. Using an electron microscope, a resolution of 0.5 nanometer can be obtained, 400 times better than a light microscope. So there are two types of electron microscope. One is the transmission electron microscope, and another is the scanning electron microscope. So transmission, like TEM for short, is used to view the internal organs of the cell, and SEM is used to view the outer surface of the cell or organism. Okay, so yeah, let's look at that. Two types of electron microscopes are now in common use. The transmission electron microscope, TEM, was type originally developed. Here the beam of electron is passed to the specimen before being viewed. Only those electrons that are transmitted pass through the specimen are seen. This allows us to see thin sections of specimens and thus to see the inside cells. In the scanning electron microscope, on the other hand, the electron beam is used to scan the surface of the structures and only the reflected beam is observed. Okay, so yeah, an example of a scanning electron micrograph is shown in figure 1.3. The advantage of this microscope is that surface structures can be seen. Also, great depth of field is obtained so that much of the specimen is in focus at the same time, and a three-dimensional appearance is achieved. Such a picture would be impossible to obtain with a light microscope, even using the same magnification and resolution, because you would have to keep focusing up and down the microscope lens to see different parts of the specimen. So yeah, it's very helpful, but we don't need to know this in details. The disadvantage of SEM is that it cannot achieve the same resolution as a TEM. Using SEM, resolution is between 3 nanometers to 20 nanometers. Okay, good pieces of information. Now, viewing specimens with the electron microscope. Figure 1.14 shows how an electron microscope works, and figure 1.15 shows one in use. It is not possible to see an electron beam. So to make the image visible, the electron beam has to be projected on a fluorescent screen. The areas hit by the electron shine brightly, giving overall a black and white picture. Like, how are the images seen? Definitely those electron beams get bounced back from the surface, and thus we can see the image. But even though they get bounced back, and those bounced back electrons can be identified, but we cannot see them with our eyes. So we need a fluorescent screen where if the electrons hit, they will be colored black color, and where they did not hit, they will form white color. So yeah, so that's kind of physics also. The areas hit by the electron shines brightly, giving overall a black and white picture. The stains used to improve the contrast of a biological specimen for electron microscope contains heavy metals which stop the passage of electrons. So why is stains used? What is contrast? Contrast is basically to differ the tone or depth of shadows of the image. So if we use staining, the electrons cannot pass through the highly dense areas, or can pass a little to the small dense areas, and thus a better image is achieved. This is again a study of the physics, students. The resulting picture is like an X-ray photograph with more densely stained parts of the specimen appearing blacker. False color images can be created by coloring the standard black and white image using a computer. So here is this diagram of how to use an electron microscope. Just look at these ones. Do not try to understand why this is happening, why this is happening. You will just know that we have to keep the specimen here. We have to use these buttons. These buttons does this work. That's it. That's all. We need to know about the specimen and the cells. To add to the difficulties of electron microscope, the electron beam and therefore the specimen and the fluorescent screen must be in a vacuum. Now there's a problem. Vacuum means where there is no air. Basically there is no air. Now there are problems in doing it in vacuum. If electrons collide with air molecules, they would scatter. making it impossible to achieve a sharp image. So yeah, if it was not vacuumed, the electron would get collided with the air particles, and thus not a sharp image would be produced. If electrons collide with air molecules, they would scatter, making it impossible to achieve a sharp image. Also, water boils at room temperature in a vacuum, so all specimens must be dehydrated before being placed in the microscope. This means that only dead animals can be examined, because the cells, the animals, they contain the water, but if they get dehydrated, they will die, and thus only dead specimen can be observed under the electron microscope. Well, that's a drawback too. This means that only dead materials can be examined. Great efforts are therefore made to try to preserve material in a lifelike state when preparing it for electron microscope. Here, look, these guys are using the electron microscope. You will learn that in great detail in other books, not in this book. Ultrasound of ultrastructure of an animal cell. What's an ultrastructure? The fine means detailed structure of a cell as revealed by the electron microscope is called the ultrastructure. We can see the structure of the cell membrane, the ribosomes, because we cannot see the ribosomes with the light microscope, the mitochondria, nucleus, chromatins. The fine detailed structure seen by the of the cells seen by the electron microscope is called an ultrastructure. Figure 1.16 shows the appearance of a typical animal cells as seen with the electron microscope. Figure 1.17 is a diagram based on many other such micrographs. Yeah, so nothing much to explain here.